Hi, everyone. Thank you very much for taking the time to hear Axcelead webinar. My name is Takaharu Hirayama. I'm in charge of our head of chemistry in Axcelead. Described in presentation title, we think that expert medicinal chemistry and co-creative research framework is quite important to establish a pathway towards success in drug discovery. So in this webinar, we'd like to introduce our unique capabilities of Axcelead chemistry, as well as co-creative drug discovery service for your research. Axcelead is a service provider supporting drug discovery and a spin-off company from Takeda in July 2017. Among our various services, our core services is integrated drug discovery service, which supports the entire process from HTS to the identification of candidate compounds. Chemistry team is deeply involved in the wide range of process, especially heat expansion, regeneration, and optimization phase. The value we can provide is a co-creation of new drug candidates through the effective DMTA cycle using our technology and experiences. And we believe that chemistry should be a flagship during the entire process. We have more than 50 researchers in chemistry. Since our establishment in 2017, we have been conducting an average of 10 drug discovery projects per year, totally 50 projects, including lead generation and lead optimization. We also have more than 20 self-developed technologies, and we will continue to develop new technologies to meet the changing needs in chemistry of drug discovery. This pie chart shows the experience and expertise of our chemists. The researchers who have been involved in the creation more than 50 candidate compounds belong to Axcelead chemistry team. Regarding therapeutic areas, we are covering major therapeutic areas, including CNS, oncology, cardiovascular and metabolic disease, inflammatory disease, and so on. As for target classes, GPCRs, receptors, kinase, and other enzymes occupy a big part of our cases. And additionally, phenotypic projects have also succeeded. Researchers who are able to address such a wide range of disease areas and the target classes are engaged in a variety of drug discovery projects on a daily basis. The Axcelead chemistry consists of multiple teams with various capability shown here in green color. Small molecule team, non-small molecule team, computational chemistry team, parallel synthesis team, analytical chemistry team, and synthetic chemistry team. Thus, Axcelead chemistry brings together the technologies and experts essential to drug discovery and co-create new drug candidates through effective DMTA cycle. In the slide that follow, I'd like to present the main capability. The first one is computational chemistry. In order to achieve rational drug design, we have developed our own guidance system named ACODE. ACODE stands for Axcelead High Performance Computing System for Drug Design. ACODE consists of more than 18 original in-situ systems and large-scale computer, including SVDD systems, LVDD systems, knowledge-based systems, and chemoinformatics systems, including AI platform. Of course, we also utilize de facto standard modeling software to design compound. We can utilize these systems in various situation drug discovery projects to provide rational drug design and analysis results for medicinal chemists and our clients. We are constantly developing new technologies as shown here. Recently, we have developed the farming system, which will be introduced later. These busy slide and the figures are representative system of Axcelead. All of these systems were developed to solve actual problem in the real scene of drug discovery. Researchers can select and use their systems in different ways, depending on their issue to try to solve it. Axcelead can be effectively placed and used in this way in the wide range of drug discovery process from target identification to read optimization phase. This is a word for example. When we want to know whether the target proteins are appropriate for small molecule drug discovery or not, this pocket can answer to the question by investigating protein surface where small molecule can bind. In the case of having co-crystal information or docking model, we often conduct SBDD approach by utilizing molecular dynamics simulators such as dynamics and MMPBSA, and thus we can determine accurate binding mode and predict highly accurate IC50 or EC50 values of compound for target proteins. This rig map is very unique systems, which is mixed solvent MD simulator to identify hotspots where small molecules tend to bind. In this system, more than 200 fragment molecules are used as a such element for finding hotspots. And then we can get a lot of information on the binding position and the binding mode of each fragment molecules. And finally, we can use it for next compound design. Additionally, we also have other unique design tools such as auto design, scaffold hopping, knowledge-based admitox prediction, and the farming system. A farming system is introduced in next slide briefly. This year, we have established a new system that guides our creation of red compounds based on the power synthesis technology. We call it the farming system, which stands for fast accessible library and rapid modifying system. We have a wide variety of original building blocks, which is one of our strengths. We have about 40,000 reagent sets. Half of them are originally synthesized building blocks, so not commercially available. By crossing these building blocks in stock, we have designed a battery compound library of more than 2 billion compounds that can be immediately synthesized. This process can automatically replace the specific moiety in the molecule of interest into an appropriate building block based on structure and property-based similarity search. At the same time, the rank moiety can also be flexibly converted, resulting in generation of various molecules different from the original molecule. Thus, this process is expected to be an excellent guidance system for the acquisition and expansion of ready compounds. The most attractive point is that we can propose many designs that can be immediately synthesized, then randomly generated. And those designs are often beyond the chemist's idea. Next, I'd like to introduce high-throughput power synthesis technology, which contributes to make a small molecule library laboratory. And it is also fully utilized in the farming system just introduced. Through this process, about 100 compounds can be synthesized within 5 to 10 days. So that quick acquisition of SOR information can be achieved. This is a list of reactions that we can apply to the high-throughput power synthesis. We can normally conduct amidation, pyridine coupling reaction, nucleophilic substitution, reaction with gases, and so on. Not only the reaction listed here, but also other reactions and multi-step reaction are also available. In addition to the high-throughput power synthesis, we can screen the reaction condition for the general organic synthesis reactions. This process can provide the optimal condition for each reaction out of 48 conditions within 48 hours. Optimized items increase type of catalyst, its reagent, solvent, temperature, and so on. The power synthesis, together with the reaction condition screening, enable to achieve high success rate and practical yield. In next two slides, I will introduce other key capabilities, analytical chemistry and synthetic chemistry. First, our analytical chemistry mainly consists of three capabilities: HPLC analysis and purification that can be applied to current compounds, and NMR analysis technologies, and X-ray single crystal structure analysis technologies. And also we can conduct crystallization screening and crystal polymorphism study, and so on. Because recently the complexity of design compound is increasing, and the handling of asymmetric compound is also on the rise. So the effective and robust purification and structure determination technologies become more important compared to the past, we think. Our strength is that these functions are gathered in one place and combined use of technology so that we can get reliable and valuable information quickly. The second other key capabilities is synthetic chemistry. The main purpose of synthetic chemistry is to develop efficient and practical synthetic route, bridging candidate compound to IND. As for bulk synthesis, we can synthesize in-house non-GLP bulks up to 1 kilogram with high quality. We also synthesize stable isotope laboratory compound for accurate pharmacokinetic analysis. But unfortunately, regarding radioisotope laboratory compound, we can not conduct hot synthesis in our laboratory. But we can propose robust synthetic route using cold compound. I show this slide again. We will fully use all our technologies introduced today to achieve an effective DMTS cycle, leading to find candidate compound for further evaluation. I expand on this DMTS cycle, and this cycle is filled with our strengths. As our strengths and obvious differentiation point from other CROs, we have enormous drug-like compounds and building blocks in our library. The number is about 1.5 million, and the fused volume of legacy drug discovery data, including specific target-related data inherited from Takeda. We can fully access and utilize these assets for your research to solve any issues. In order to strongly support Russian drug design and in situ study, we have self-developed computational chemistry systems, A-code, and we also have productive synthesis technologies, including high-throughput parallel synthesis, and their purification, structure, determination technology, and so on. Practical synthetic route scouting and scale-up synthesis and many experiences are quite helpful for your drug candidates to move forward for further development. Regarding evaluation technology, we of course fully equip evaluation platforms, including screening, biology, DMPK tox, other than chemistry in one place. By combining our strengths with client technology, data, and experiences, we think we will accelerate co-creative DMTS cycle. This slide shows our standard goals and optimization period for medicinal chemistry. Typically, we will confirm PK and PD response in vivo as goal to be achieved in lead generation. We assumed a standard period of 9 to 12 months for lead generation. In lead optimization, we aim to not only meet the criteria for the in vitro activity and acceptable tox properties, but also achieve sufficient in vivo PK, PD efficacy in appropriate doses and administration route. The standard period is expected to the 12 months. We will flexibly change the goal and the period according to the client's requirements and depending on the situation of the project. And we promise to develop an optimal research plan and execute it. To give you an idea of how to proceed with actual co-creation project, we have prepared a Gantt chart like this. This is very similar to the Gantt chart of actual drug discovery project. In the co-creation project, the client and Axcelead will cooperate closely and execute their project together to achieve the goal in the shortest period by setting up an optimal division of responsibility that maximally matches the strengths of each party. For example, the client can transfer their assay system to Axcelead, and Axcelead can be responsible for the admin tox evaluation, while the client conducts their proprietary biology evaluation. For the chemistry, we can also divide the chem types between the client and Axcelead. Of course, we can also optimize their same chem type together. We have a lot of experience in the co-creative drug discovery research with many pharmaceutical companies and other groups so far. For example, our collaboration with these green-colored companies are announced on our website. Also, we have a lot of achievements in each drug discovery project. And we can introduce the case of Cordia Therapeutics as one of the representatives. Cordia Therapeutics is also known to be a spin-out company from Takeda. They announced that they entered MORT1 inhibitor CTX177 and the related compound into a license agreement with On Pharma. Actually, our chemistry team had conducted this project and strongly and directly contributed to identify candidate compounds CTX177 as project leader and members. Let me summarize my presentation. Our medicinal chemistry team has 4 strengths compared to other CROs from the viewpoint of co-creative drug discovery research with clients. First is that we can provide rational drug design based on deep insight of target and in-depth data analysis of various properties of compound. And we also utilize unique A-code or original guidance system I introduced today. Second is that we have advanced synthetic chemistry capability, including high-throughput power synthesis, developed through our long experience in drug discovery. Third is that we have rich success and failure experiences and deep knowledge in drug discovery. And we can access legacy drug discovery data and enormous compound library. Finally, we can totally provide high speed and high quality DMT cycle because there are all functions, including chemistry, screening, biology, and DMP tox in one place, leading to cross-type with each division in Axcelead. Okay, we are very happy if you are interested in Axcelead and Axcelead chemistry, even a little bit after listening to today's introduction. And we are looking forward to collaborating with you if we have a chance. Let's start co-creative drug discovery with us. That's all my presentation. Thank you very much for your kind attention.